My name is Humaira Afrina, 206467.
Hi everyone, my name is Anusna, 207630. Hi everyone, my name is Nursaida, my metric number 206674.
My name is Ang Jiaming, metric number 206504.
My name is Shira. My metric number is 206175.
Hi, I'm Rashida Durrani binti Muhammad Nazari. My metric number is 207181.
My name is Afif, number metric 205784.
Hello, my name is Said, 208241.
In this slide, we will answer all the question provided by Dr. Sarah. So this is the background of it. Okay, so I will just go right into our first question, which is the steps, the experiment and the steps for microarray. So this is the basic principle of DNA microarrays, which lies on the hybridization between the nucleic acid strands, and the property is such as the complementary nucleic acid sequence will specifically pair with each other by forming hydrogen bonds between complementary nucleotide base pairs. So like for the example provided here, samples will be labeled using fluorescent dye, and at least 2 samples are hybridized. Complementary nucleic acid sequence between the sample and probe attached by hydrogen bonds, and then the fluorescently labeled target sequence that bind to a probe will generate a sign, and then the presence of at least one cDNA sequence in thousand sequence can be detected. So before we start our microarray DNA microarray, we will need to prepare our probe. First, we will need to annotate the genomic structure, and then we will PCR to amplify the probes, and then continue by spotting the PCR products into the microarray tray. Then I will label it in here is as tray 1, and then next we will get right into the microarray experiment. Next. So after we prepare our probes, we will need to prepare our sample mRNA, which we want to confirm whether it has Taxol, and then the reference that contains Taxol mRNA, which is confirmed has the Taxol, the mRNA Taxol. Okay. So first thing we need to do is reverse transcription and then label the mRNA with fluorescent dye, and then as you notice there's a probe attached to the mRNA. And then you color both samples. The sample one you color by green, and then the reference that has Taxol mRNA is colored by red. And then we will mix it into tray 1, which we prepared the probe earlier, and then hybridize. And then by hybridization, we'll use the excitation laser and then scan it, and then you'll get to analyze the image. So how do we know whether the sample has Taxol or not? On the right side, I have put the indicators. So the significant part is the green one, the sample that shows a green sample will be only presented in a sample only, and then the red is reference contained in the Taxol only. And then the yellow one means there's both colors, both mRNA in these 2 mRNA, like that. So yeah, I think that's the overview of the experiments. Next.
So how to confirm the gene involved in Taxol production? So first we can use based on result of cDNA microarray. So the result will be shown with different color of fluorescent dye. So for further information, we can use BLAST by obtain the FASTA format for the specific gene and run it use BLAST-P. Related and similar sequence will be shown involve what species the sequence from. So this is example by using BLAST-P. So this is BLAST using FASTA format of DB80, but from different species. So this is example of gene involved in Taxol production like TS and DB80. So I think that's all. This is a gene that, for example, gene that involved in Taxol production.
Okay, next, we perform quantitative PCR. So in this experiment, firstly we prepare reaction mixture, qPCR master mix, forward, reverse primer, and probes. After that, we transfer the mixture into the wet plate. Next, the cDNA sample, which is Taxol brevifolia, is added, and the well is covered by microscopic film. Then we run the program in real-time PCR machine, and lastly, data is analyzed.
Yeah, after real-time PCR is conducted, our target sequence of, for example, Taxodione synthase sequence present in the cDNA sample of Taxus brevifolia will be hybridized within the sequence, and fluorescent signal will increase. Hence, it can use for detection of genes involved in Taxol production.
Next, I will explain the molecular beacon. So basically, molecular beacon is a single-stranded oligonucleotides in the form of hapten structure, and the function is to increase the specificity of the real-time PCR. So molecular beacon consists of a stem that contains nucleotide sequence which complement each other and are connected to the quencher and fluorescent. Next, a loop complementary to the targeted DNA sequence, and the quencher at the 3 end and fluorescent at the 5 end. In a relaxed state where there is no targeted DNA present, the nucleotide in the stem region will remain bounded, which causes the quencher and the fluorescent closer to each other. So in this position, the fluorescent is quenched, thus no fluorescent will be emitted. when the targeted DNA is present, the loop of the molecular beacon will change its conformation to linear strands and bind to the targeted sequence. So this causes the quencher and the fluorescence to separate and unquench. So when it is unquenched, it emits fluorescence and allows the DNA polymerase to attach to the targeted DNA and extend it. So once the extension occurs, the molecular beacon will detach from the targeted sequence and return to its original structure.
So for the advantages and disadvantages of molecular beacon, here's the list. For the advantages of the molecular beacon, the probe are preserved during the reaction. It increases the specificity and sensitivity of the reaction. It also increase the thermal stability and efficiency of hybridization. And also the molecular beacon facilitates the detection of multiple targets using multiple fluorochromes in a single reaction efficiently. And lastly, it has greater cycling flexibility and optimized parameter. For the disadvantages of using a molecular beacon is to require a relatively large complementary target sequence to ensure that the complex is stable, and some of the non-specific interaction of hairpins can lead to false positive. It's a bit expensive and it's require a design for probe.
Okay, so for difference between result obtained by real-time PCR and normal PCR. So real-time PCR, for your information, the short term is not RT-PCR because RT-PCR is reverse transcriptase PCR, but the short term of it is qPCR, which is quantitative PCR. So the difference between the result between by a real-time PCR and normal PCR is real-time PCR technique that allows the detection, characterization, and quantification of nucleic acid for various applications, while normal PCR is a technique that allow analysis of a short sequence of DNA by amplifying selected segment of DNA. Real-time PCR or qPCR accumulate the amplification product. The accumulation of application product is measured as the reaction progress or in the exponential growth phase, while the normal PCR, they measure it at the endpoint analysis or plateau. Next, real-time PCR product can be detected in each amplification cycle, while normal PCR, the product, which is the amplicon, can be detected by the agar-residual electrophoresis. Real-time PCR need less time, is less time consuming, and the results are precise. This is because we measure it at the exponential phase, and it is the optimal point for analyzing data, while the normal PCR is time consuming and result may not be very precise because the results are based on size discrimination, so which may not be precise. So that's all for this.
So next is Q&A session. First question is how MJ increase the yield of Taxol?
So I think how MJ increased the yield of Taxol is by having the Taxol as a secondary metabolite. And then secondly, it will accumulate at low level. This is because part of the defense response of the plants against the pathogens. Pathogenic attack. And lastly, it elicits the MJ to trigger and activate the response.
Yes, true. For your more information, MJ can be used with the low energy ultrasound to increase the yield of Taxol. So the yield will be higher compared to using them individually. Next, I pass to Durrani.
So for the next question, what will happen when the probe is hybridized within a target sequence in the steps of real-time PCR?
I think the answer is fluorescent signal is produced.
Yeah. Fluorescent signal is produced when the probe or molecular beacon is hybridized with the target sequence.